Let's start the fifth class of sixth week. So, in the last class I introduced you and we talked extensively about the density gradient centrifugation, and if we go back to the class you will realize that I told you that most of these work achieved on adult hippocampal neuron around 1995 to 2000, then between 2000 and 2003 much of these technologies were translated for spinal cord neurons. I told you around 1980s embryonic motor neuron separation followed density gradient centrifugation. So, there also the logic was same, because when you look at the spinal cord of embryonic motor neurons, so the way the cells are being separated, if you look at the spinal cord, so the spinal cord is something like this. In an embryonic and as well as in the adult. So, when you see the animal from the top, you get a dorsal view. When you see it from the bottom, say for example, okay. So, you are seeing the animal from the top. So, this gives you a dorsal view. If you see the bottom, this gives you a ventral view. Now, if you look at the spinal cord is in this animal, it will be like this, or say for example, this is human. So, if you look at the spinal cord from this side, you open the viscera and look at the spinal cord, you will be looking at the spinal cord from the ventral side. Whereas, if you look it from the back, you look it from the dorsal side. Okay. So, keep that concept in mind. So, it is very interesting, the spinal cord is kind of like this. Just like you have done paper folding, you fold this wherever I drew the line, you fold it. So, what you get something like this then, right? After folding, you will be getting something like this. So, this is a kind of a central part, this is the side and this is the other side. Now, these two sides fold outward. In other word, these two sides, if this is the ventral horn, if this is, you are looking at me at ventral side, and if I keep it like this, so it will be folding only like this. So, you see my arms on both sides, try to look at it like this. If you look at my arms on both sides, this side and this side, so those are the green. So, this is the part where both my, this is where both my arms are. So, this is the red part. Now, let me just show you out here, that will make more sense. So, this is the red part like this, and this part of the arm, this is what you see. This is the blue shaded region. One second. This is the blue shaded region, and it folds like this. So, it seems that the blue part is as if on the top. It is the same structure like this, it curves like this, and this is the blue part, the one which I am shading now. So, now if you want to culture motor neurons, you have to know the location of the motor neuron, spinal cord motor neurons. So, the location of the spinal cord motor neurons are here, which is called the ventral horn. But now, these cells along with the motor neurons, they are of two types there. There will be alpha or there will be gamma. So, the differentiation does not happen so early, but in the embryonic phase, but it happens in the adult. But the alpha are the ones which are the biggest one, and the gamma are the ones which are the smaller ones. These are smaller size, these are larger size. Now, apart from it, you have a series of, if you have the alphas here, you have the gammas somewhere out here, you have a series of other cell types which are present here, which includes your oligos, residing microglia, and oligos or oligodendrocytes, residing microglia, astrocytes, and there are some along the central canal, which is essentially a location like this. Just at the center, you have a lot of stem cells. Now, again the same problem, if you remember, except now this is no more adult hippocampal neuron I am talking to you. Now, this become embryonic motor neuron culture. And what you have? Alpha motor neuron, MN, gamma motor neuron, possibly very small neurons. Then you have oligos, you have microglia, residing microglia, you have astrocytes, and series of them. And the technique which is followed, which was followed, was the same. And this was the very, very first paper. If I could dug out the paper, I will definitely circulate it. They form a density gradient column, and on a density gradient column, they separate out these cells. This was the very, but before you do so, you have to first take the embryo and you have to dissect out only this part. And once you dissect out this part, which is called the ventral horn, then you dissociate the cells of ventral horn, and these cells then are being separated using density gradient column. On a density gradient column, you separate out these cells. Once you separate out these cells, what you will be achieving is something like this. On a density gradient column, the largest cells, which are the motor neurons, they will form a wonderful layer, if you are really good at it, a small thin layer out here, which will be the pure motor neurons. And if I remember it right, this was done with sucrose as well as with nicodens. And there are specific devices which are being used to measure the density. I will give you those papers there, you can check it out, what are the kind of devices which are being used to make these kind of density gradients columns. Okay. So, these pure motor neurons, which are the embryonic motor neuron, EMN, embryonic motor neuron, are the source of studying motor neuron disorders from embryonic system. And again, the process is density gradient. But there is an advancement to that process which happens. That's what I wanted to share now, which is immunoseparation of cells. Immunoseparation of cells is, first of all, try to understand the basic fundamental before I can. So, a cell is a dynamic system. It expresses a whole sorts of receptors on its surface, and a cell distinguishes itself by the kind of receptor it is expressing. Now, say for example, during the development of the cell, if you know that at a specific time point that X or Y cell is going to express a specific receptor on its surface, then if you have a technology by virtue of which you can grow antibodies against that receptor, then in a pool of different cell types you can separate out that cell. Well, let me draw it, that will make more sense. Say for example, I have these different cells, same size, but they are of different types, I know that. How I know they are of different types? I know that these cells have receptors like this. I am just showing the color coding, that way it will make more sense to you. And these are external receptors. Okay. So, we see green colored, blue colored. Now, I give you a situation, I say to you, okay, give me the green label or green cells. And I believe these green cells are, say, motor neurons. And this process in the development may be a very unique phase. So, I know that say at E14 motor neuron expresses this receptor, but after by E16 or E17 there will be again a mix and match is going to happen. But if I know this is a zone where there is a unique situation that these motor neurons will be exclusively expressing a receptor, and this is where I always say, if you want to become a good cell culturist, you should always keep a tab on the progress in development biology. This will be always helpful. If you understand development, or if you can keep a tab on the different development taking place in development biology, it will be very helpful to make very judicious judgment. So, at around E14, there are motor neurons. Most of the motor neuron expresses a very unique receptors. I am just telling the story how people went into immunoseparation of neurons, motor neurons, as a case study. Now, what they did was very interesting. Parallelly, there was some individual who developed earlier than that an antibody against this part, the one I am circling. Antibody against the extracellular domain of that receptor, domain of that XYZ receptor. This was already known. Now, somebody did a very smart thing. You love what they did, and I will send you the paper, then you will realize what they did. They took this antibody, okay. So, they know this antibody, and what they did, so you have a dish. On this dish, so I am labeling the antibody with, say, this color. They coat the dish with this antibody, okay. Now, dish is coated with the antibody. Now, what you do, you take this mixture of cell and you roll the mixture of cell on top of it. So, what will happen? The cells, if they are having the receptors for this particular thing, they will go and bind. and other cells which are the red cells, blue cells, pink cells, they all will be moved out because they do not have an attaching antibody to it. Right? Now, this is what forms the basis of immunosuppression. It's a very simple concept, but you just have to get this idea, and that's why I'm kind of drawing in different colors. But catch, you really have to know your cell type or your concerned cell type, does it express that antibody or not? And in motor neuron biology, some very smart moves which happen, and I'll give you the papers which will kind of give you those unique papers which really change the way the field has progressed. And there are very discrete events which took place, like E14 motor neuron expresses a particular extracellular receptor. This was discovered by a gentleman called Johnson in St. Louis, Missouri, back in 1980s probably, or earlier than 80s. Earlier to that, there was one gentleman, I have forgotten his name, who for a totally different reason, absolutely nothing to do with it, developed an antibody against the extracellular domain of it. And then there came a third person who had an idea about these two. What he did was creating a test bed like that, and they could get all the motor neurons separated from all the rest. It's a very smart technique. And then what you do, then you needed to have a competitive binding. You have to remove these. There are two ways you can remove. You can tap it very hard. If you tap it, then this attachment is not very prominent, and in order to achieve this, you need almost 30 minutes. You have to keep them on a dish. After 30 minutes, what you can tap them, and these cells will detach. Or what you can do, you put some excess antibodies there, and which will kind of competitively will bind to this to the receptors and will remove these cells, and you can collect them in a you can collect them in a different centrifuge tube in a buffer medium, and you can spin them down and you have a pure population. So, this forms the basis of immunosuppression of motor neurons. Isn't it a cool technique? So, we started with, now think of it where we started. So, first what it requires a proper dissection ability, step 1. Then it needs enzymatic dissociation, and then it requires density gradient centrifugation. D stand for density, G for gradient centrifugation. Then from there you can do a immunoseparation. You see all these different steps by virtue of which you can get a very, very pure population of the cell of your choice. So, this is how you achieve these very interesting feat. Now, if you go back in the last lecture of 5th week, now do you feel more learned that given your situation, I do not know, each one of you have your own set of situations, you should be able to use these different techniques to achieve the goal. And here I must mention you something very critical in the last lecture. So, this whole thing is a very, very time dependent one. Unless this is an E14 spinal cord, you won't see this pattern. By E15 the pattern changes. So, that is what I say, that you have to have an understanding of developmental biology, or at least you should keep a tab how the development is happening, so that you can separate out things. You know exactly this antibody or this particular receptor will be there. I can utilize, I can capitalize on it. Okay. So, this is how most of these things happen. They happen, it is not there is any plan, there is any planning, that is how it happened. It just happened to be like that. Okay. So, with this, I will close in this lecture with the density gradient and immunoseparation. Next week, our goal will be to understand the 2D and the 3D cultures, again with a special reference to neurons, which will kind of help you to appreciate how the whole field is moving. Okay. Thank you.